you have 3 nucleotides that span 2 codons get removed from the DNA. And so when these 3 nucleotides get removed, it causes the deletion of an amino acid called phenylalanine at position 508 in the CFTR gene. So CFTR is this protein that your cells need, and we'll get to why it's important in a little bit. Basically, you just lost one amino acid in this entire protein that is going to end up having these big issues. So you lost it, but your transcription factors bind to the promoter regions on the DNA exactly as they should. You've got RNA polymerase come down and link onto that template strand, and it makes the coding strand version of the RNA. That RNA gets processed just like any other RNA, perfectly fine, where you get that methyl guanosine cap added to it. You get the introns removed via the acceptor, donor, and branch sites in those introns. The spliceosome links together those 2 exons, and you also get that poly A tail put onto that mRNA exactly as it needs to in order to go out into the cytoplasm from the nucleus. So everything's just fine so far. And in the cytoplasm, you're gonna have a ribosome grab onto this mRNA that's floating around, and that ribosome is gonna begin to translate this protein, that sequence of nucleotides, the mRNA. And as your ribosome is sequencing and translating this protein, the first few amino acids on that N-terminus are going to contain what is referred to as a signal sequence. And so these signal sequences are going to have things called signal receptor particles recognize it. And so as your ribosome is making this protein, a SRP will come and stick onto that specific signal sequence that it's identified and has affinity for, and it will carry the ribosome plus the mRNA plus itself down to the endoplasmic reticulum. And on the endoplasmic reticulum, you have something called an SRP receptor, a signal receptor, a signal sequence receptor, protein receptor, particle receptor, sorry. And so basically, once you get this onto the endoplasmic reticulum, the SRP receptor is going to cause the actual remainder of the nascent polypeptide chain that's getting sequenced to go into that lumen of the RER, the rough endoplasmic reticulum. And so as this protein is being synthesized in the lumen of your RER, you're going to have something cleave off that signal sequence to get rid of that, because that's not actually needed for the final product of this protein. So that happens exactly as we would expect it to. And then finally, you get the primary sequence of amino acids completed, and this protein will begin to take shape and it will begin to fold. And the issue is, at this point inside of our endoplasmic reticulum, that we lost that phenylalanine, and because it doesn't have a phenylalanine, the protein doesn't fold exactly the way that it should. And because of that, it exposes an inner part of this protein that shouldn't typically be exposed. And because of this exposure, you have something called ubiquitin, and ubiquitin has affinity for regions of proteins that shouldn't be showing. And so ubiquitin ligase is this other protein that's basically going to come by, and it's going to link a bunch of ubiquitin onto this misfolded CFTR protein. And specifically, this is for the nucleotide binding domain, the NBD1 domain specifically. And so you've got this misfolded protein with a bunch of ubiquitins on it. And as soon as this thing leaves the endoplasmic reticulum and enters the cytoplasm, because it's got this ubiquitin tag on it, it's going to have a thing called a proteasome form around it. And a proteasome is basically a blender. It's going to take this misfolded protein and just chop it all up into its amino acids and completely delete it. And so that happened to NBD1 because of that phenylalanine mutation. But the rest of the protein subunits that make up the CFTR protein, because the CFTR protein is composed of multiple subunits, it's referred to generically as an ATPase binding cassette. And so it's got these other subunits. And so basically these other subunits get made just fine, and they get put into the cell's membrane. But the issue is that because they didn't get that one subunit, the NBD1 subunit, because it was misfolded and degraded in the endoplasmic reticulum, they're not able to function the way they're supposed to function. And so as you have this one amino acid deletion, those 3 nucleotides, because of that, your cystic fibrosis that CFTR protein is not functional. And so why does that matter? So with that CFTR protein, that ATPase binding cassette, usually what's supposed to happen is that you have your N binding domain 1 and 2. And so if the cell received a signal to open up this thing, it's basically just a big gate. If it receives a signal, it opens up this gate, and you have chloride ions inside of that cell that are supposed to be able to leave because of this new gate that they have that's open for them, and they can't do that anymore. So in a normal healthy cell, what's supposed to happen is that you would have a receptor tyrosine kinase get activated inside the cell, and then it's going to phosphorylate it's going to bind to the CFTR protein. It's also going to attach itself and attach phosphates onto this protein to stabilize this active state where the gate's open for those calcium ions to come through. But because we don't have that NBD1 anymore, because of that phenylalanine deletion, it's not able to open up the way that it typically would, and we don't get chloride ions leaving the cells that we typically want them to. So the consequences of this is that cells called epithelial cells that make up ducts or glands in your pancreas are not able to make mucus the same way that they should be able to. So the lack of these chloride ions being able to leave the apical, which is just a way of saying the top part. So if this was the lumen, or this was the duct up here, and then we had our actual epithelial cell right here, the apical membrane is this part, the interface between the actual cell and this duct, and that's where you have these CFTR proteins are supposed to allow chloride ions out into this thing to make a nice, non-viscous mucus. And so because they're not able to get these chloride ions out, the mucus that is being secreted by glands is way more thick than you expect it to be. Consequently, this will make things get clogged up. And so a lot of the enzymes that your pancreas would typically secrete, like lipases or proteases, or the bicarbonate that's needed for neutralizing the stomach acids in that small intestine, are not able to get out because the whole duct is just jammed up with this really thick mucus because of those chloride ions not being able to get out, because the CFTR protein itself was mutated in that NBD1 subunit with the 3 nucleotides getting deleted that remove that phenylalanine. And so because of all this stuff, your pancreas isn't able to get these digestive enzymes into your small intestine, and that means that at the end of the day, the patient is going to be very malnourished because they don't have the lipases to break down the lipids, they don't have the proteases to break down the proteins, and they don't have a way of effectively neutralizing the pH of the stomach. And because you can't maintain a good pH in your small intestine, the other enzymes that are involved in digestion are not going to be working as they optimally should, because the charges are going to be different on the R domains of those proteins. And so because of all these things, malnourishment is the key symptom or issue that patients with cystic fibrosis have. And so the way to treat patients with cystic fibrosis is to have them take these supplementary digestive enzymes with every meal, so that as that food is in their stomach and in their small intestine, it is able to be in a form that you can have cells in that small intestine called enterocytes actually take and use the carbs, fats, and the sugars from the foods that were just consumed. So that's how you treat cystic fibrosis. Now, it's very important to treat it as quickly as possible and to recognize it as quickly as possible, because with cystic fibrosis, a huge problem is that as soon as the baby's born with cystic fibrosis, it's being malnourished. As the baby's feeding itself, it's not able to digest things that other babies are able to digest. And so the more time that goes by that cystic fibrosis isn't diagnosed, the worse it will get, because the body is being deprived of all these nutrients that it typically would have and needs. And so that's a key thing here. And then a final thing I'm going to say with cystic fibrosis is that this is an autosomal recessive disorder. What that means is that it's something where if you had a parent who was a carrier and they mated with another parent who was a carrier, they have a 1 in 4 chance of having a kid, boy or girl, doesn't matter, but a 1 in 4 chance of having a child that will have cystic fibrosis. And so knowing the genotypes of the parents will be very helpful, and also doing a pre-screening before the baby is born will also be extremely helpful, because if it is identified that the baby has cystic fibrosis, you can get on top of this way more quickly and make sure that the baby has the digestive enzymes that it needs. I will say also that cystic fibrosis is much more common in white people than in other races, and there's speculation as to why cystic fibrosis is as prevalent as it is, because 1 in 3,400 is a pretty high number. And so the reasoning or the hypotheses for this right now involve explanations surrounding the heterozygote advantage, which is to say that if you are a carrier for cystic fibrosis, but you don't actually have it because it's an autosomal recessive, if you're a carrier for it, then that could make you more fit to your environment. There were some studies that showed that having or being a carrier for the cystic fibrosis mutation ended up giving people better resistance to cholera. So you do get an advantage out of this if you're a carrier, but if you're homozygous recessive, then that's when you have cystic fibrosis, and we need to treat that immediately. And then the other explanation for why is it as common as it is because people are suggesting that the CFTR gene itself may have a very high mutation rate. There might be something that we don't know of that causes those 3 nucleotides to very easily get taken out from that DNA that we haven't thought of, or it's just a very common occurrence. So these are possible answers to why cystic fibrosis is as big of a deal or as common as it is. So I hope that this video is helpful and interesting. I hope you guys are all doing well. Let me know if you have any questions, and thank you all for watching.